Good evening, everybody. A very warm welcome to our AOZ Foundation. And here we are once again to give you a treat of three beautiful online knowledge based lecture series on account of our AOZ Foundation Day 2023. Tomorrow, 19th of December, we have AOZ Foundation Day. This is the fourth year for our organization, which is working immensely for zoology and animal sciences. So, once again, greetings for everybody. And at the end of this year in December, we hope that you will have this 3 days retreat as a takeaway of your final 2023 and utilize it in 2024 for making your careers and professional life much more enhanced with the talks and the knowledge information that would be shared from this platform. So, friends, let me tell you something about AOZ. AOZ is a non-profit organization. Its aim is to work for the progress, knowledge dissemination of zoology, animal science, to bring up and provide a platform for young zoology people so that they can work in the field in a much more better way. We are open to collaborations with other agencies, organizations, universities, institutes who are into higher education and academics related to animal science, zoology, and various other allied fields of this particular subject. If you are willing to become members, please do write to us. We will be sharing soon the email IDs and the further details of AOZ. We also have our social media accounts. We are available on Instagram, YouTube, and we are also having Facebook accounts. You can follow us over there. This 3 days online knowledge retreat. Today is the first day wherein we want to talk about autophagy and cancer by bringing a very eminent expert onto our platform, Dr. Mayur Sayani. Do join with us tomorrow as well at the same time, and also on 20th to have the retreat related to different topics which would be useful to you for your career further. So let's not take too much of the time, and let me invite our today's guest. Good evening, sir.
Yeah, good evening, Neil, ma'am.
We are very happy, privileged, and obliged to have you on our platform with such a short notice. You have always been extremely supportive for students, and we really look forward to the talk that you are going to deliver today.
Yeah, thanks, Noel. Thanks a lot. And yeah, today I would be talking about the autophagy, and my talk is designed basically for mainly for BSc students and uh, a very early career, even after 12th standard. If students are in even in they are in the first year, I want to introduce to them. So it is very basic, but at the same time, it is very exciting. And I encourage, if students are there and they want to stop me and interact in between, ask the questions, they are free to do that. I am more comfortable actually in offline mode, where it is highly interactive, where students ask the question, pause me at the right time and ask the question.
Yes.
So they are free to do that. And I would love to come sometime in Surat and deliver a talk there and interact with the
Surely,
students.
surely,
Yeah,
sir, surely. So,
yeah.
friends, sir would like to have questions and your interactions into our comment box. So, please keep sharing when we start with the talk. Before I actually start and no welcome him to deliver his today's topic, let me introduce him in a better way. So, sir is basically very eminently working in the field of no cell and molecular biology. He is presently assistant professor at Vikram Sarabhai Institute of Cell and Molecular Biology, MS University of Baroda, Gujarat. You would be happy to know that he did his MSc in biochemistry and thereafter he did his PhD in medical sciences from Tokyo Medical and Dental University, Tokyo. And he is the one who has a long and extremely intensive research experience by doing postdoctoral studies at number of institutes. Firstly, to name his postdoctoral no, studies, he did it from Tokyo Medical and Dental University. Thereafter, he was a postdoctoral fellow at Molecular Biology and Genetics Unit, no, Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. He was also a postdoc at National Center for Biological Sciences, Bangalore in 2017, and also at IISER Bhopal in the year 2018 to 19. He has a lot of good publications in international reputed journals like Cell Biology International, Journal of Clinical Investigation, Journal of Applied Polymer Science, Journal of Cell Biology, Autophagy. So he is the one who is extensively studying and is an expert of autophagy, which you should really listen to. Now, apart from that, Sir has been awarded a number of awards and honors, like he has been awarded a Lifetime Lectureship Award by the Council of Scientific and Industrial Research National Eligibility Test. He also owes a prestigious MEXT Fellowship from Japan government. He was awarded a prestigious Global Center of Excellence Fellowship again from Japan Society for Promotion of Science during the year 2012 and 13, and Tokyo Biochemical Research Foundation also gave him a fellowship during 2013-15. Now, he owes a grants, number of grants. Presently, he's working on one of the projects by DST SERB CRG, which is funded almost 42 lakh rupees, and that is about dissection of crosstalk between autophagy pathway and NRF2 axis during starvation. He has been awarded number of grants in the past as well. He was, I think, he's also working on one of the research project sanctioned by Research and Development Cell under the title Effects of SARS-CoV-2 ORF3A and ORF7A on host autophagy as in principal investigator. So, friends, we put forth a person who is really expert in the field of autophagy, and I hope you all will take enough advantage of our today's guest, and there would be lot of takeaways for you today, so that you can enhance your career and get motivated that there is lots to work in the field ahead. So, sir, wonderful to have you, and I would not grab your more time but allow you to be with our people who are waiting to listen to you. So please carry forward your talk.
Yeah, thanks, Nehal ma'am. And I will present my screen. And if any technical problem is there, do let me know. We will solve it and
Surely.
we go further. Yeah.
Surely. So I'll provide you more space. So you might not see me on the screen, but I'm there in the green room. So
Sure.
you and your no the online screen wherein you want to work off, that would get more space on the board.
Sure. So my screen is visible, right? You can see the PPT.
Yes, sir.
Yeah, Neha ma'am, can you see the PPT? Can you confirm?
I think you need to add to the stage. So should I add it for you?
Yes, can you please add it?
Yeah. Yes, sir. It's visible now.
It's visible? Yeah, yeah. So, hi, this is Dr. Mayur Sahani, and I'm assistant professor at Dr. Vikram Saraf Institute of Cell and Molecular Biology, the MS University of Baroda. And today I would be talking about the introduction to autophagy and cancer. And as I said, uh, it's very preliminary introductory topic, and I will also discuss the reason why it is so. And uh, I will go very, very slow. So even the first year BSc student, Bachelor of Science student can also follow it up. And uh, I will also mingle the some of the messages which my mentors and my supervisors during my career gave me, and that time they sounds very simple, but then it was very, very effective throughout the career. And uh, there we go. Yeah. So y- the cell has a synthetic machinery, for example, protein translation and mRNA transcription, or RNA transcription for that matter. And if I take the right color to choose, yeah, we have, yes, we have synthetic machinery. At the same time, we have degradative machinery also within the cell. And there are two type of degradative machinery. One is very selective, as it says, which is ubiquitin proteasome machinery, which degrades protein. Another one is the autophagy, which is bulk degradation system. So let's see the selective autophagy, selective proteasome system, which degrades protein selectively. And in this cartoon, a protein is shown, and that is ubiquitinated one over other, and then it can enter the 26x proteasome, and then it releases the building blocks. If we go in the more detail, how it is done. So let's say we have a substrate here, red in color, circle red. What is the substrate? So substrate may be aggregated misfolded protein. So let's assume in this cartoon it is a misfolded protein. Then the question is how it is selectively recognized by the system to be degraded by proteasome system. It is selectively ubiquitinated by set of enzyme called E1, E2, and E3 enzyme. These are ubiquitin ligases, and that selectively ubiquitinate the misfolded protein in this example. And there are many of them, hundreds of them in a representative cell. And once it can be ubiquitinated in variety of way. So for example, here it can be polyubiquitinated. The red color is the misfolded protein, and these blue small circles are the ubiquitination. And if one over other ubiquitination happens, they are called polyubiquitinated. It can be at here it can be multi-ubiquitinated as well. In this cartoon, where monoubiquitinated ubiquitination happens at different places, and it can be monoubiquitinated. But the destination of all this marking of ubiquitination is the degradation by proteasome system. It's called 26 proteasome tunnel-like structure. The misfolded protein enters in the tunnel. Inside the tunnel, we have proteases, variety of them, and then it chop off the protein. And what we get is a building blocks. What are the building blocks? Amino acids and peptides. This system of protein degradation is known, is very well given in the textbook, and it is elaboratively covered in the syllabus also. However, autophagosome, which is an organelle like any other organelle, like mitochondria nucleus, this organelle is not very well covered up to the best of my knowledge in the standard textbook of BSc and MSc. In fact, in one of the standard book, I think Bruce Albert's, only half a page about the autophagy, and that surprised me that the early career researcher at the bachelor level, they should be introduced to this wonderful topic, and it's very essential for the cell. Therefore, I decided that in the Gujarat region, the west part of the India, I will sensitize people for this a very important topic, and therefore it is very preliminary, very basic, and I try to make it exciting. Okay, if I have a next slide. Yeah. So, as Nehal ma'am introduced me, that my PhD is from Japan, and I stayed there for almost 6.5 years. So, a long experience there. And during my doctorate degree, I published this paper. Let's not go into the detail. It was a autophagy paper, and I'm the first author there, and this is my supervisor and only one postdoc. And the postdoc has helped me to mentor my project. So all the work is done single-handed by me, and that's how the culture of the lab. So I told you I'll give you a few message also. So what I got here in this tenure is a solid rigor training, and all the experiment in this paper was done by me. And this paper was so relevant that it is cited till today 377 times. And there is the famous Mount Fuji of Japan, and this is the cherry blossom. In colloquial language, they say it's Sakura, and they make a very famous tourist destination using this cherry blossom tree. And one of the destination was, in the stretch of 1 kilometer line, both the side of the canopy is just by this cherry blossom tree. And actually, we can do here also. While I travel from Baroda to Ahmedabad, from Baroda Express Highway, you can see variety of Keshuda flower and the lavender green stretch of that. And sometimes I think we should make use of this plantation of indigenous tree. It can be converted to the a tourist destination. Okay. So here it goes. While I was there, roughly around 7 years, I happened to meet this great scientist, Professor Osumi. Is a Nobel laureate 2016, and his student was Professor Noboru Mizushima, and his discovery of one of the important factor of autophagy was appreciated throughout the world, and in fact that made him Nobel nominee as well the same year. And so Professor Ozumi, Nobel laureate 2016, then his student Professor Mizushima, and then Professor Mizushima is actually my supervisor, and I'm standing here at the Dr. Vikram Sarabhai Institute of Cell and Molecular Biology, and these are my students, basically my lab people. So basically, these the four generation of researcher are depicted in these slides. Now, we used to meet. Of course, Professor Mizushima, I was in his lab, so every day we had some or other interactions. Professor Osumi was not in the same place, of course, in the same country, but at the different place. So we have yearly interactions, and it was great to interact with him. I will share a few of his messages in this lecture as well. So this is the zoom version of the same picture, and then what I want you to notice is that he's holding one frame, and this is basically the EM picture of the yeast cells. What you are seeing, this spherical circles are basically the yeast cells. And he often talked to us during the yearly gathering. His famous advice to all the researcher, regardless of their age, whether they're senior or they're junior or younger, that read every day, work every day, and what you work, report every day. And he said that repeat this cycle almost every day. You don't have to stress out, read little bit, work little bit, report what you have worked. And to the lab leader, he used to say that choose the work which you like. And he used to tell us many times, almost every meeting, if he said, professor, what is the advice for the new new lab or a new researcher, he often say the same thing, and it was very accurate. And we used to meet in a trial lab where three lab, almost around 100 people get together, and they discuss their 1 year data, and then without overlapping and with the healthy competition, we used to share our data, right? So, and he actually showed us that choose the work which you like. And if I go to the next slide, yeah. So he was passionate, and he used to tell us that I like to observe the cell under the microscope. And these slides depict the same thing. His lab work on the yeast, and this is the yeast cell. He simply like to observe the cell under the microscope, Saccharomyces cerevisiae. And what he did in his work, this is in 1993, yeah, here. So he looked, as he said, that do the work you like, and he liked to watch the cell, observe the cell under the microscope for his scientific curiosity, of course. And in this slide, you have wild type yeast cells, and on the right hand side you have APG1 mutant cells. APG1 is one of the autophagy pathway genes. So what happens in this typical yeast cells, you can see a little bit shadow. These are the vacuole of the yeast cell. So vacuoles are the lysosome like structure in the yeast cell where they chop off the material and they have degradative enzymes. What you don't see in this picture is the autophagosome. So autophagosomes are very small, and then these autophagosome deliver the material to the vacuole to be degraded. And the word itself suggests autophagy. Uh, how the word is coined, basically, auto means self. Self, my mouse is not doing great, but and phage is for eating, degradation, right? So when the self is eaten by the cell, it is called autophagy. So when the wild type cells are starved for the nitrogen, they need to have amino acids to synthesize the protein. Therefore, what they do, this cell where autophagosome we cannot see, but they capture the cytoplasmic material and give it to the vacuole. In the wild type cell, this process is very efficient. But if you see the mutant cells, you can clearly see this vacuole is brighten up. And as you increase the starvation, more number of hours in the presence of PMSF, which stop the degradation within the vacuole, you can see the clearer and clearer, bigger vacuole. And these are the empty vacuole, because now autophagy cannot deliver the material into the vacuole. However, in the wild type cells, where autophagosome has delivered the material to the vacuole to degrade, right? And therefore, it is called autophagy, self-eating process, and the genes involved in autophagy is called ATG, autophagy, ATG genes. And then he published in 1993, and this is just the one protein, one factor, which is protein in nature. But later he and his group, another group in across the world, let's say Kleonsky from USA and other people, they find out several mutants and therefore several genes of autophagy. And the way to do it, you look under the microscope and you look for this kind of cells. If this kind of cells which has empty vacuole, empty clear vacuole, you can send it for sequencing, and the gene which is mutated, that can be find out, right? So classical genetics, you do the random mutagenesis on the yeast cells, look for the phenotype. What is the phenotype? Empty vacuole. And once that is there, you send it for the sequencing. You find that this gene, let's say, yeah, ATG, one of the gene, ATG1 is, yeah, it's already there here. ATG1 is responsible for this phenotype. But let's say if this pathway has more number of genes, you will find more number of gene also in your mutagenesis. Then what was found out, the in a yeast cell, more than 30 genes are found to be responsible, directly uh, involved in autophagosome formation. And don't bother about the number, and don't try to remember them. The message is that autophagy is self degradation, where the vacuole in the yeast, which is a degradative place, the autophagosome deliver the material there. And to make the autophagosome, there are more than 30 autophagy genes are responsible. That is the message. And these genes are categorized in various complexes. There's ATG1 complex and another complex, and ATG12 conjugation system, and 8 conjugation system. You don't have to remember. Get the gist, get the concept. So, by classical yeast genetics, having a phenotype and a great curiosity, we found out the autophagy genes, more than 30. Everyone is okay with here? Neil ma'am, everything is so far so good?
Yes, sir. All well.
Yeah. Okay. Yeah. Okay. Then while Professor Osumi was doing this research, my supervisor, Professor Noboru Mizushima, joined his lab as a postdoc. And and he was interested in mammalian cells autophagy, whether mammalian cells has a autophagy, and is it different from yeast, and uh, there are, or whether autophagy has more functions in the mammalian setup. And what he found during his tenure in 1998, Professor Mizushima, in 1998, under the supervision of here Professor Osumi, and he is the first author, he found out that, okay, let me show you the data, what he found out. So, you know that I have showed you that yeast picture, uh, where in the APG1 mutant, the vacuole was clear. So, Professor Mizushima dealt with another ATG protein. Let's say that time it was a nomenclature was APG also. So ATG and APG is basically same. And before, every scientist, autophagy scientists across the globe, decided to call it ATG. 2 3 nomenclature was there, and uh, the scientists used to call ATG APG as well. So what he did was, he took the wild type cell, and this lane is a basically immunoblot. And for a BSc, very young, first year BSc student, the immunoblot is basically, you can tell the level of protein by the signal, right? So this is the immunoblot signal, and you can tell this HAAPG12 is present in this much amount. So if you take the wild type cells, and this is anti-HA immunoplot, that means HA is hemagglutinin, and uh, APG protein is tagged with the HA tag. And because of this HA tag, now you can use the anti-HA antibody and recognize this protein. So while Professor Mizushima was doing this immunoblot, and alone HAPG12 monomer is around 30 kilodalton, what he saw was one additional band. And typically they label as a star asterisk, and the asterisk generally stands for the non-specific band. So he thought it's a non-specific band because it was not there in the wild type. So something HAPG12 is giving non-specifically, because molecular weight of APG12 was around 30 kilodalton. However, when he extended his curiosity and do the same anti-HA blot, uh, what he got to his surprise is basically, uh, okay, yeah, I remember. This is not anti-HA blot. This is rather the anti-APG12 antibody blot. So what you get here is APG12 signal, and in all the lane you can get APG12 signal. However, you get this non-specific band only in one of the mutant and wild type. So what does it says is that APG12 is somehow bound in the to the some factor, most likely protein, and it has a higher molecular weight. And these are the protein or genes responsible for making this connection between these two, APG non-specific band and APG12. Later he figured out in this uh, very highly cited, more than couple of thousand times this paper is cited. What he found is that indeed that APG12 is connected to this non-specific band. And what is that protein? It turns out that this is the monomer of APG12, which is shown here. And what is this so-called non-specific? It turns out to be very specific. It turns out to be APG12 is bound to APG5. And because now this APG5 has additional molecular weight, you get a huge molecular band shift above in this Western blot. And these are the enzyme which was catalyzing this reaction. So if any one of that factor, which is protein in nature, are not there, you will lose this binding of APG12 and APG5. So he discovered this, and that this ATG12 is conjugated by covalent bond, and because of the covalent bond in SDS page, this interaction is still holding together. And this conjugation of ATG5 and ATG12 actually catalyzing another conjugation of LC3. What is LC3? Another protein. What is this protein doing? This protein is also catalyzing autophagosome formation. This protein also has a conjugation, like ATG5 is conjugated to ATG12 by covalent interaction between glycine and lysine. LC3, another small protein of autophagosome biogenesis, is conjugated to lipid phosphatidyl ethanolamine. And we will see in the further slides that how this is important for the autophagists across the world. So basically, Professor Osumi find out the many yeast ATG proteins, and during that time, a contribution from Professor Mizushima lab and others across the world find out the similar homologue counterpart of the autophagy in the mammalian system, and more or less they were doing the same function, but in mammalian system they have additional uh, additional functions. Okay, so I just give you the glimpse starting from the yeast vacuole to the autophagy, but if you see the actual biogenosis with many factors involved, what you see is that the cell has to decide, the signal has to come that hey, induce the autophagy, and that comes from amino acid starvation. It passes on to mTORC1. I will explain everything. And then one more complex, and in this example it is shown on the ER, but other group has also showed it on that autophagosome membrane comes from mitochondria or the plasma membrane. And here is the autophagosome formation starts. And what you should notice is that this autophagosome is not sealed yet. It is immature. The sealed autophagosome mature autophagosome is depicted here. And if you notice, there are many factors on immature or unsealed autophagosome, ATG 12, 5, or 16L, all these VP, PI3P, all these factors are not present in mature autophagosome. That means this mature autophagosome, while it is going to fuse with the lysosome, all these proteins are going to be spared by the lysosomal degradation, and that's how the system is very, very smart. In a stressful condition like starvation, you don't have to invest in translation of this protein. So in during the biogenesis, these ATG proteins are used, but before that autophagosome gets sealed, all of them go away from the mature autophagosome, and what is going to be fused with lysosome is mature autophagosome, which has only few proteins. One is LC3, few are there, adapter proteins are there, and few others. Okay, so the question is, I told you the one of the signal can be amino acid starvation, but what are the other upstream regulators of autophagy? And the regulators are basically one of the very important regulator, upstream one, is called mTORC1, mammalian target of rapamycin complex 1. And this protein is basically master regulation that decides to go for cell growth or not to go for it. It takes inputs of stress, level of oxygen, whether the cells are in a hypoxic condition or normoxic condition. It takes input of growth factor, amino acids, energy, amino acid starvation, energy depletion, whether there is plenty amino acids or not. This molecule can sense, and one can wonder how a single protein molecule can sense so many stimuli. And the answer is very simple. One is the protein is very big and they have lot of domains that, and it binds to so many other proteins. So therefore it justifies that this complex is big and have a variety of proteins that binds to it, and therefore it can act logically, can act as a master regulator. So we have studied, I give you the glimpse about the autophagosome and its historical view. And so what are the physiological roles of autophagy? Uh, and to our surprise, actually this is the cleaning machinery of a cell, self degradation during the stressful time. And therefore this autophagy is used in variety of physiological function of the cell. So for example, the autophagy degrades the cytoplasm and damage organelle and generates the amino acid. So during the starvation, amino acid can be generated. During the development, autophagy is required. So for example, in the Dictyostelium discoideum, if one of the autophagy gene is mutated, the Dictyostelium will not make the fruiting body. This is the part of the development. It have anti-cancer effect that I will cover in last two slides. It is anti-aging. So nowadays, the intermittent fasting. What happens in intermittent fasting? The person is actually inducing autophagy, right? So and there is a caloric restriction when the mice were kept on caloric restriction, not nutrient restriction, caloric restriction, and they live very, very longer. Their lifespan increases significantly. Therefore it is anti-aging. It is involved in cell death and survival. Autophagy is also involved in antigen presentation. So when your APCs, antigen presenting cells, and otherwise when the MHC of the immune cells, and they have to present the antigen on the surface, the autophagy chop off the antigen and then help in flagging that up, that this antigen is there. Therefore antigen presentation is also dependent on autophagy. Bacterial degradation. if there is an infection, if not all, many pathogen and bacteria can be identified by the autophagy machinery for the adapter and other recognition machinery. And this bacteria is literally engulfed by the membrane of autophagy and then fuse it to the lysosome, and where the pathogen can be destroyed. And if there is a stress of protein misfolding or proteotoxic stress, those fibrils or the protein aggregates can be cleared by the autophagy in intracellular clearance process. So for me, maybe I'm exaggerating, but then for me it is as important as glycolysis, right? And it has so many functions. And if you see the number of publications, they are in the in tunes of thousands, where every year more and more research is showing us the fascinating role of autophagy. Now there are the questions. For example, what is the time scale of this autophagosome formation? So basically this question is how much time it takes for autophagosome formation till it fuses with the lysosome. How many autophagosome can be generated in a cell, and at which location inside the cell they are generated. So this slide I will use to answer those questions. So from uh, autophagosome is basically a double membrane structure where it enwraps the damaged organelle like mitochondria, it can be nucleus also or part of nucleus, and of course cytoplasm. It enwraps this material and make a mature autophagosome. This mature autophagosome fuses with lysosome. Once it fuses with lysosome, the hydrolytic enzymes of the lysosome chop off the material inside and then release the building blocks. The entire process by the live microscopy from Mizushima lab is around 10 to 15 minutes. It takes depending on the stimuli and the cell line you use. And how many autophagosome can be generated within cells? Hundreds of autophagosome can be generated in the span of 1 hour or less than 1 hour. And where what is the location of autophagosome generation? Initially, there are few schools of thoughts. Initially, people used to believe it is at ER, but the later it has been shown that the material of the autophagosome generation, especially membranes, can be supplied by mitochondria and plasma membrane as well. So we know now in a great deal about the kinetics of autophagosome. So the next question is, autophagy is always non-specific. That means amino acids are not there in the cell, so the autophagy can simply go and engulf the part of cytoplasm, fuses with lysosome, and bulk amino acids are released within the cell. And remember, when you are starving or fasting, let's say right now, for example, if you are having a dinner, then it's all fine, you have plenty of nutrient. But let's say if you skip your breakfast, lunch, and even dinner, you think you are fasting, but your cell is not fasting. Your cell immediately undergo autophagy in tunes of minutes, and then it generates the amino acids for the translation of very essential protein, right? So the question is whether autophagy is non-specific. It just a bulk degradation of a random material in the cytoplasm. Uh, it turns out that
So, Mayur sir,
yes.
there are a few students, it seems they have joined in between the lecture. So,
Hmm.
can you just give a gist of what you have explained till now so that they can have a little understanding just in few lines?
Yeah. So far, what I suggested, or rather explained, is basically how the curiosity-based science was driven. Professor Osumi, 2016 Nobel laureate, through the yeast genetics, found out the the genes involved in autophagosome formation, or Autophagosome is an organelle just like your mitochondria and ER. It's a double
Hmm.
membrane structure. It can be induced during the stressful condition at the basal level. Few autophagosomes are always there, and it is the mammalian autophagy was studied by many scientists after Osumi, and it basically degrades the cytoplasmic material and damage organelle, and it can also degrade the pathogen and generate the amino acid, and it's a cleaning material of the cell basically. Yeah,
Yeah.
yeah,
Thank you
yeah.
so
Okay.
much.
Yeah,
So I
yeah.
hope, sweetie, you have got the gist of what we are talking, no, during this lecture, and you can revisit the YouTube channel to have again the view of what he has been explaining. Yes, sir. Please continue.
Yes, correct. So the question was whether it's a non-specific, bulk, and random. Then yes, the answer is yes. When if it is needed, it is non-selective and generate the amino acid. But if it also can be specific, so for example, this the P62 is an adapter protein which binds to the autophagosome through LC3, and it's bind to the ubiquitinated protein. Now in the first slide I showed the many misfolded proteins are ubiquitinated and are degraded by proteasome. So if they bind to the P62, and they do bind, they also can be degraded by autophagy, right? So this is the selective autophagy where misfolded protein can be degraded by autophagy. And if this process is not there, selective autophagy is not there, you have a aggregate formation inside the cell which is toxic. You have NF kappa B activation, you have unnecessary apoptosis, and you can have a unnecessary NRF2 hyperactivation. Another selective autophagy can be the, let's say your mitochondria is basically damaged, and you know the mitochondria is the generation of ROS, reactive oxygen species, by the mitochondria is highly controlled. But if the mitochondria is damaged, the ROS will be throughout the cytoplasm. Therefore, this damaged mitochondria can be sensed by autophagy machinery, and again the autophagosome can engulf this damaged mitochondria and clear it off. And while damaged mitochondria is identified and cleared by the autophagy machinery, it's called mitophagy. If the ER is damaged, then it is called ER phagy. If the nucleus is damaged, it's called nucleophagy. And to our surprise, if lysosome is misbehaving, it is called lysophagy, right? So basically, in the real sense, it's a vacuum cleaner and a destroyer also, where any damage to the cell is to the organelle can be cleared off by the selective autophagy. And if, let's say, bacteria infects the cell, then this bacteria can also be identify the autophagy machinery, adapter proteins like optinurin, NDP52, P62, and this bacteria can also be degraded. And if we don't have this autophagy machinery, the infection can flourish inside the host. So these are all schematic and all, but how in real cell or autophagosome look like? And it look like this. This is the in the in the plastic tissue culture plate. I am showing two cells, and this is mouse embryonic fibroblast under the microscope. And this is the fat condition. That means they are not in any stress. They have plenty of amino acid, growth factor, and glucose, and these cells are basically stained for LC3. What is LC3? I explained you before. It is small protein marker of autophagosome. They are found on mature autophagosome. So in the growing condition, this is the basal and if you see this puncta, this these are basically signal of LC3, and we tell they are proxy for autophagosome. They are on autophagosome, but this signal is basically implies that these are autophagosome. And if you starve these cells, let's say you starve them for 2 hours, what you are starving for? You are starving for amino acids and growth factor. What happens is number of LC3 puncta is increasing. Therefore, you can conclude that number of autophagosome are increasing. And if you zoom this, actually not zooming, if you look the same thing under the electron microscope, this one dot is basically is this kind of structure, a double membrane structure, and this autophagosome is not sealed yet. Therefore, most likely it has other molecules like ATG12, 5, 16L, and uh, all those molecules. It This is mature autophagosome. It's completely spherical. The membrane is sealed, and it should have LC3 on its membrane. So this is the way, using LC3 immunofluorescence assay, observing them under the microscope, you can see the autophagosome. Another way to judge whether the cell is undergoing autophagy induction or not, you can do the Western blot, immunoblot. You can use the adapter protein P62, anti-P62 antibody, and if you see the level of P62 protein in the growing cells, starvation hour 0, that means there is no starvation, that means plenty of nutrient amino acid there. This is the level of P62 protein. But if you starve the cells for 1 hour or 2 hour, then autophagy is induced and P62 protein is degraded inside the lysosome. And if I give even the broader version of EM where other organelles are visible, you can definitely see the autophagosome here and this double membrane structure. It is not sealed yet, but once it fuses with lysosome depicted here, you can see many autolysosome by this electron dense structure are basically the autophagosome has fused with the lysosome, and it gives you the electron dense structure by the EM, electron microscopy data. And this cell is basically mouse ES cells, and they were starved for 2 hour amino acid starvation. So the next question is, how would you monitor the autophagy? You want to say that the cell or animal is undergoing autophagy or not. What are the markers for autophagy induction? So that you can, of course, you can do the immunoblot for P62, which I just showed. You can also do the immunoblot LC3. I told you that this is the growing condition. That means plenty of nutrient, and you have this LC3 signal. And this is the basal level of LC32. LC32 is basically protein that is lipidated. And the more this LC32 form, more the lipidated LC32 is there. That means more the autophagy. Therefore, when you starve the cells, here, this line, what you see is that amount of LC32 form increases very significantly. And further, this increase LC32 accumulate further if you don't let autophagosome fuse with the lysosome. So what happens is, if you stop this fusion, this LC3 inside the cell will increases, and it is LC32 form because you have induced the autophagy by starvation, right? So this is called LC3 flux assay, and this is called P62 monitoring the level of P62 to monitor the autophagy. So during my tenure and stint in Mizushima lab, I was monitoring the autophagy in the tissue culture plates, and then I this is the basal level of P62 protein, and then if you starve them for 1 or 2 hours. So I set up the experiment for 1 and 2 hours, and then I went outside for some other work, and I did not plan this time point, but the outside work basically got stretched for another 6 hours, and then I starve my cells for 8 hours. So imagine for 8 hours, the cells are under tremendous starvation of amino acid and the growth. And then I came back and I was like, oh my God, the cells are starving for 8 hours. I supposed to do it only for 2 hours. I had two options in the night. I go ahead, collect the lysate and do the Western blot. next day, or and when I'm not hopeful that till 8 hour of starvation the cell will even be there, or a healthy cell would be there or not. But then with curiosity in mind, I go ahead and did this western blot, and then what I found is the P62 is basically restored back life, just like the growing condition. And I was surprised. I thought I made some mistakes, so I repeated this experiment of 8 hour starvation. That time nobody does 8 hour starvation for monitoring P62. The entire globe most likely is doing the 2 hour starvation, and I repeated three, four, 5 times, and every time I got the same result. And therefore, I decided to take this as my PhD topic, and then we dissected what are the factors that govern this P62 restoration for very long time starvation. It turns out that this P62 is restored back for long starvation of 8 hour, and that restoration depends on autophagy. So you need intact autophagy for uh, P62 restoration. You need transcriptional upregulation, and you need amino acid. These amino acids are generated by autophagy. And therefore, we give a caution to the world that this people used to uh, monitor autophagy by looking at the P62 level is down or not. Then we give the caution to the world that uh, one has to be very careful while interpreting the P62 levels, because if you give a long time starvation, the P62 level uh, uh, completely restored back. So this was 2014, and by now it is cited hundreds of time, and it's a very fine piece of work. So this is so far about autophagy, and the I thought in whatever summary form, in very preliminary form, I could convey to the students at the BSc, early MSA, it would be great. And then again I am going to deliver this kind of talk, and people become more aware about autophagy here. And as my talk says, uh, I want to touch upon cancer also, but the cancer is a very popular topic. Almost everybody knows what is cancer, but we know cancer in terms of the genetic mutation. But without genetic mutation, can you think about a cancer? Uh, and that just a very short 2 slides I want to show that thing in context of autophagy. So the cancer cell is basically it's a very fine balance of it can go towards tumor promotion, and the cancer cell go towards tumor suppression. And autophagy can play a dual role. It can promote tumor, and it can also promote the tumor suppression. If I give you example of a, let's say, tumor suppression, then this is the liver of a mice, and these are the mice basically are autophagy deficient mice, tissue specifically. So where autophagy is depleted in the liver, and this suggests for the month. So here 6 month autophagy is depleted, here 9 month, and here 19-month autophagy is depleted. So in these autophagy depleted mice, as the time passes, the reactive oxygen species generated, and that become the cause of liver tumorigenesis. But in the wild type mice, which is picture is not shown here, the liver is completely normal. So this data clearly suggested that without autophagy, liver is going to get the cancer. So it suggests the tumor suppressive role of autophagy, right? And if I give the tumor promoting role of autophagy, I can use this cartoon that in the solid tumor where the cancer is established, the tumor is established, and inside the tumor there is a lot of competition, cutthroat competition. Competition for what? Nutrients. And if I use this cartoon inside the cell, and you can see the blood vessels and blood capillary are not reaching much inside. So inside the cell there is a scarcity of nutrient, glucose, amino acids, vitamins, oxygen. And in such stressful condition, the cancer cell use actually autophagy to survive. So in this tumor, if you block the autophagy by, let's say, chloroquine or other inhibitor, this this tumor cell has a hard time to survive. So the autophagy plays the dual role in the progression of cancer. And I will stop here. I wanted to give the glimpse of that, and I'll come up with more such knowledge based the lectures. And I acknowledge here my knowledge is uh, you know from variety of places, including the lab in uh, Japan, Professor Mizushima lab, my one of my exposure in Ravi Manjitha lab at JNCSR Bangalore, in NCBS Bangalore. I was in Vardarajan lab, at Pushkar Sharma lab during early days, New Delhi. And my teachers at MSU, while 2002, while I was studying biochemistry, they put the foundation of the fundamental knowledge in me. The funding comes for sub TST, and Max fellowship is from Japan. If somebody wants a guidance how to get this fellowship, I will be happy to help. And these are my lab people, and interns, dissertations, and PhD students are welcome to apply. And interestingly, my lab people come for the short time, also very short time. So here we have Purna, and now she is doing PhD at Boston University. She was in my lab for 2 to 4 months. This very recent picture of my lab, well, Janvi was there in my lab for around 4 months, and now she is absorbed for PhD at NCBS cluster at INSTEM, and rest of the people are dissertation and PhD students. So feel free to contact me for short term or mid-term training in my lab. And thank you very much.
Thank you so much, sir, for such a wonderful and timely, talk related to autophagy and cancer. And it was really a very simplified version of a great intensive work that you have done it. I'm sure people will appreciate this. We have a lot of viewers no onto the YouTube, and this would roll on now. So there would be a lot of other students all over India and abroad as well who would take advantage of this particular talk of yours. I have two questions to put forth to you. One is, if somebody wants to initiate know himself or herself in autophagy research, from where they should begin. No, as in student, if I have an interest in the subject, how I should start. That's the first question. And what are the recent updates in terms of research now, which they can be taken up in autophagy?
Yeah, so very relevant question from student point of view. And then, so I think the one of there are several ways, and one of the best when your student is read about that subject. So read about autophagy if you're interested in autophagy, and gather enough knowledge. And in the field of autophagy, the problem may be the papers are overwhelming. So the select the pioneer paper, and there are very good scientists in India as well. Santosh Chavan, Ravi Manjitha, and so many others. You can read that paper and then contact us. We have a small group of autophagy scientists in India, so you can
Okay.
contact us and then clear your doubt. And then if you're really passionate about studying autophagy, you get some training in their lab for 2 months during summer or your dissertation. And you can just come for short term. So currently those things are available in my lab. And then you be in environment where autophagy is discussed, autophagy is researched. It's a vast topic, and then you are good to go. Yeah.
Yeah. And what are the updated research topics which are now being worked upon in autophagy?
Yeah, so basically the host pathogen interactions and the that is one of the very hot topic, and then inflammation basically, which Santosh Chavan lab is doing, and
Yes.
yeah, and in the cancer and autophagy is very hot. So because it is a double-edged sword, actually where the autophagy plays role, and at in which setting, at which time point of the cancer progression, that is really very very hot topics currently going on.
Yes, and those who are interested in immunosciences, immunology, inflammation is a very talked about topic nowadays for number of diseases. So,
Yeah.
autophagy could be one of the channels you can work upon with respect to inflammation. So, try your hands onto that. And those who are really interested, because I know there are so many students who are, looking forward and get attracted to work in cancer biology. This would be one of your lifeline to start your cancer research and take it forward. So please look into these, read as it was advised by the Nobel laureate, whom we started the slide with. And so last question before we end up today's session, what would be your advice for the faculties who are into teaching, how they should take this particular topic forward in their classes, or with the experiments that they might be involved in the curricula?
Yeah, so for the faculties, I suggest that they can pick up any latest review, and they have a generally they have very good teaching experience, so they can read and definitely understand and share it with the students. That is one. In the lab, I suggest that they can do they can start thinking about in their model system, or in their research field, whether autophagy is playing any role or not, right? So many of the work is done like that. So somebody has zebrafish or somebody has C elegans, and then they are studying, let's say, the behavior, and then one day the idea pop up that, hey, this behavior, or this neuronal circuit depends on autophagy directly or indirectly. Then they can simply contact the autophagy scientist, and it can be quickly checked. So basically it's a initial collaboration, a small collaboration is needed, and then that's how the collaborative work is done, and there is no hassle. People do help each other, and it's
Yeah.
great going. Yeah.
Yeah. So wonderful to know note these points. I'm sure everybody is taking notes of what are your advices and the guidance that you provided today. On behalf of AOZ, no, Association of Zoologists, I really is passing on the gratitude and a thank you note towards you for making the presentation so lively, simplified, and bringing the new topic onto our floor today, because such topics are very rarely discussed so openly, and a very niche area which is now open for everybody to look into it. So once again, thank you so much, sir, and thank you everybody who have been with us today for this particular session. Please stay tuned for two more these kind of very informative, retritive, knowledgeable sessions coming up for you tomorrow as well as day after tomorrow. Tomorrow we are going to have Ajay Baldania. He's going to discuss how to deal with the bibliography that every researcher at every level is finding it difficult to handle when they are into their dissertations or their research projects or doing PhD. So he will be talking bibliographic, no, in context to that, bibliomatrix. So please stay tuned with us. Do join us on our YouTube channel for tomorrow as well. All the life members of AOZ Foundation, we are going to announce your life membership no, online tomorrow. So please do stay with us tomorrow for that. Also there would also be presidential no address on account of AOZ Foundation Day. So, Mayur sir, once again, thank you so much for joining with us, and we'll keep
Thank you.
looking forward to you for this kind of talks in future, even for the offline talks. Thank you
Yes,
so
yes,
much,
ma'am.
sir.
I will really love to interact in offline mode, and it can be hybrid as well. And thank you
Yes.
very much for inviting and the audience. Thank you very much for your patience and opinion are all mine. It may be partially incorrect, the advices and all, but I thought I will share it anyway and
No, no,
can motivate some.
it was indeed wonderful to go through a very, very niche and important area, which is to be looked into by scientists. Thank you so much. Thank you,
Thank you, ma'am.
everybody. See you tomorrow. Good night.